Thanks, Rick, for the introduction and for having me over today to talk about spinal muscular atrophy, a rare neurological disease which is a bit of a success story. Genetic cause of it has long been known, and as of a year and a half ago, the first initial therapy was approved in the United States. However, that success story comes with some caveats and some limitations that I think provide important lessons in drug development also for other diseases. So what I want to do today is to tell you a little bit about spinal muscular atrophy, or SMA, as I will mainly talk about. Tell you what type of disease it is, what were the reasons that SMA was a disease that where therapy development has been relatively successful compared to some other rare neurological disorders that are notoriously difficult to treat, and tell you some of those limitations and what we can learn from them. So SMA, I take it that most of you are not very familiar with this disease, sometimes referred to as floppy baby syndrome or Whitney-Hoffmann disease, by the German neurologist that discovered it many years ago, is a disorder in which motor neurons, the nerve cells that control muscles in the spinal cord, degenerate specifically. And that leads to weakness of muscles, and therefore mainly children that have the disease never develop normal motor skills such as sitting and walking. It's the most common genetic cause of death in children, and it affects roughly 1 in every 10,000 newborns. There's some variability depending on what country is looked in. Although there is one specific genetic cause, there's a fairly broad variability. The most common type of the disease, roughly 50% of all patients, it affects very young children, babies, but it can also occur later in life, even up to disease onset of 40 years or later, in which case it's not nearly as severe a disease as it is in young children. So what is very important to discuss is the genetic cause of SMA, because it forms the basis also for how therapy for this disease was discovered and developed successfully. So everyone has this chromosome 5, which is one of the 23 pairs of chromosomes that humans have. And on this chromosome 5, there is this stretch of DNA that's a bit unusual, in the sense that through evolution, it got duplicated and inverted, and several copies of the same genes are present there now. What I'm focusing on today is these two genes, SMN1 and SMN2. And both those genes lead to the production of a protein called survival motor neuron. And it's the survival motor neuron protein that is essential for all cells and tissues to function properly. What happens in SMA is that one of those genes, the SMN1 gene, is completely missing. That's the definition of SMA. So SMA is diagnosed genetically and characterized by the absence of this gene. Luckily, there is a second copy of this gene called SMN2, but it has a problem. It's slightly different from SMN1, very slightly different, and that small difference towards the end of the bit of DNA that codes for the survival motor neuron protein leads to the protein that is being produced to be unstable and quickly degraded. And only a small minority of this SMN2 gene leads to the production of a full-length, stable survival motor neuron protein. So what does that mean? It means that if you have not a lot of SMN2, you have a very severe type of the disease. If you have more SMN2, you have a much milder type of the disease, which is indicated in this table, where very young babies that die soon after birth only have 1 or 2 copies of this SMN2 gene, whereas patients in which the disease starts later in life have 4 or more copies of SMN2, and therefore more survival motor neuron protein. What has been very helpful in developing treatments and finding treatments for SMA is that we have, in the lab, we have available a number of good animal models. There's no way around it in basic science. When you want to test out various novel therapies, you'll need reliable animal models to test out those therapies. You can't go straight into patients. And so those mouse models in particular, and they're based on this genetic defect that was identified in patients. And when this genetic defect is replicated in mice, they develop a type of disease that more or less resembles the condition in humans. And that is that those mice don't grow and develop normally. So very quickly after birth, they fail to gain weight, whereas here in the black line depicts healthy mice. The gray line depicts the SMA-like mice, and as you can see, they fail to gain weight compared to their healthy brothers and sisters. And when we look at how they move about, it's a very simple little experiment that you can do with these mice. By putting them on their back, they are very keen to flip over again. As you can see, the healthy mice do that really quickly after birth. Within a week, they flip over almost instantly. And the SMA-like affected mice can't do that. And that reflects the trouble they have moving around because their motor neurons are degenerating as they are in the human condition. We have a number of other tools as well, including zebrafish, which are a relatively new model. And zebrafish, of course, they're tiny tropical fish that are very far away from humans. What you can use them for is the first few days when they're developing, they're transparent, so you can see right through them. And so what we do is we make these motor neurons fluorescent, and then we can follow the development of motor neurons by investigating the zebrafish, by looking at them with the microscope. What you can see is if you, again, mimic this genetic defect that occurs in patients, the motor neurons that are, the cell bodies of the motor neurons are up here, and the nerves that go out to go and control the muscle movement of those zebrafish, they don't develop like they would normally. So this is actually a very powerful tool to test out compounds, therapies in a very quick way to see if you add another gene or another drug or another whatever compound you're interested in to those zebrafish, do then the motor nerves develop normally again. So a bit of the time course of how SMA therapy development has been. As with many diseases, I suppose, the initial description was made many years ago, at the end of the 19th century, and then it took essentially more than 100 years for the specific genetic defect to be identified. And after that, it's been a relatively quick development in the SMA field. I say relatively quick, but we're still more than 20 years later than the initial discovery of the genetic defect. But that genetic defect led to the initial development of a number of animal models that I showed on the previous slide. And those animal models then allowed for a number of different therapeutic approaches to be tested in those models that went on into clinical trials round about this 2011 time. That is the end of this particular slide. This is from a paper that is not by me, but I really like the figure, and I'm finding an excuse to update it at some point to make it go up to 2018 at least. So, in summary, so far, SMA has been characterized by a number of really nice opportunities for therapy development. For me as a scientist, it is really good if I can investigate a disease that has a single genetic cause that makes it straightforward. So I can focus on one gene and the function of one gene and how it affects the different disease models. These are particularly robust in SMA. They reflect a number of important hallmarks of disease that we also see in patients. It doesn't matter in which part of the world you investigate, and we're always very reliable and reproducible. And perhaps most importantly for therapy development is that although this SMN1 gene is missing in all SMA patients, they all have a second backup gene. And although it's faulty and it's not quite as efficient as SMN1, it's there, and therefore it can be therapeutically targeted. On top of that, in the SMA field, there have been a couple of other really useful things going on in the background, really, that have really sort of supported the field and improved the efficacy in drug development. I think are particularly nice to discuss today, also in line with the collaborative things that Rick just mentioned in his introductory talk. So talking about collaboration initially, what has worked really well in the SMA field is that although it's a rare disease, it's not super rare. So, for example, in the UK, there's roughly 1,300 people that are affected by it. And so there's been a reasonable amount of interest from scientists around the world to work on this disease. And somehow, this wasn't, I don't think this was ever formally discussed initially, but people ended up working on their own niches, on their own small bits of interest. And so science is always inherently competitive, but somehow in the SMA field, because everyone had their own little points of focus, this was very complementary. And that has been very efficient. And so to illustrate that, this gives you an idea of various groups in Scotland that are working on SMA, and they're all focusing on different aspects of the disease. So, for example, our colleagues in Aberdeen focus on tissues other than motor neurons that are affected in SMA. We've worked for many years with people in Dundee who are experts at a very specific a molecular technique to identify proteins that are changed in SMA. St. Andrews has a very good cell biologist who works on the molecular mechanisms that are linked to the survival motor neuron protein. And Glasgow, we have someone who works with zebrafish and is also a good cell biologist. And then me and my colleagues in Edinburgh, we focus on a number of different techniques to sort of combine all those insights together and come up with new therapeutical targets for SMA. This is also going on UK-wide, and this was formalized in the UK SMA Research Consortium that was sponsored by the SMA Trust, a patient organization for spinal muscular atrophy in the UK. And again here, this consortium was very much based on local expertise, people that were leaders in their field internationally, not necessarily all working on completely different topics, but very much collaborating on advancing their own skills, advancing their own knowledge. And so the SMA Trust has funded one postdoctoral researcher in each of those places, in each of those universities. And the consortium meets regularly. It discusses their findings, exchanges protocols, particularly, for example, stem cell research. Many different protocols to work with stem cells exist, and in order to increase reproducibility and reliability of those experiments, it's very important to work together on how you develop those protocols and how you can exchange them between different places. So this has been going really well, and it's been going on for the last few years now. And also within Europe, there's an umbrella organization called SMA Europe that represents all the local countries' SMA disease charities and funds research in Europe. And these are just some examples of various places that I and the others in Edinburgh have collaborated with over the years, but there are many more people working on SMA throughout Europe and also in the States. And I think this has been instrumental in getting to a stage where it was possible to develop therapies for this disease. So this is another thing that has really helped the drug development process in SMA, is that this is of course, a very simplified black and white version of classic drug development that most of you will be familiar with. So a scientist finds something in a lab, a pharmaceutical company takes it forward, develops it further, gets it into a clinical trial, and eventually, if all goes well, it gets submitted to the regulatory bodies such as the Food and Drug Administration in the USA and the European Medicine Agency. And then if all is well, the drug gets approved. So that's not really how it's been going in SMA research, and probably also not how it should ever be going. Again, it's a simplified and black and white way of showing it. But in SMA research, particularly the American Cure SMA charity has very strongly advocated research into SMA, and has always had very close ties to the regulatory bodies that need eventually to approve the drug. And how it went was that they organize an annual meeting to which they invite scientists from around the world, to which they invite pharmaceutical companies. So we all fly out to the States once a year to discuss our latest findings. Then Cure SMA collates that, discusses that with the FDA, says, well, this is how it's going. What do we need to do to maximize our chances of getting approval for this drug? The FDA feeds that back, and then the patient advocacy group feeds that back to the scientists and the pharmaceutical companies. And so there's been a very successful sort of interaction between all these different stakeholders to eventually lead to approval of this one drug for SMA a year and a half ago. So I've mentioned this drug many times. So what is the idea behind it? Again, the figure that I showed very early in my presentation, the idea for the therapy is very simple. We have this SMN2 gene that is very similar to the SMN1 gene, except for one small change towards the end of the gene. How can we make this shortened, unstable gene product? How can we increase the proportion that is actually a full-length stable gene product and leads to full-length protein production? And with maybe some excessive detail here in this part of the figure, but I think around 2010 it was that people in the United States actually discovered a bit of DNA that is close to this part of the SMN2 gene that's usually missing, and it leads to this protein product being unstable. And they found that when you block this bit of DNA sequence, that increases the proportion of this bit of the SMN2 gene that gets included into a full-length version of the protein, and therefore leads to full-length survival motor neuron protein production. And so it's actually, although it might look complicated, the idea is very simple. There is a short bit of DNA, 20 of these letters. If you block it, you increase the full-length SMN gene product. Obviously, it was more complicated than that and getting developed and everything. But once it was ready for a trialing in patients, they found that this drug called nusinersen, now marketed as Spinraza, quite significantly increased the overall survival in SMA patients. And so these graphs on the left side represent very young SMA patients before the age of 2. The graphs on the right side represent older children between 2 and 12 years of age. And both show that the overall survival was almost doubled in patients that were treated with nusinersen after a year. And in somewhat older children, their motor capacity, their motor skills were much improved compared to the untreated groups. So what is this drug? So long word, antisense oligonucleotide, just to say a short bit of DNA that targets the other bit of DNA, and it leads to higher survival motor neuron protein levels. It's complex because you can't just take a pill with a bit of DNA and get it to the right place. In this case, because it needs to go to motor neurons and it needs to go to the brain, you have to inject it straight into the central nervous system. And so that's complicated, of course, because a lot of these children are very young and it's not so easy to do those lumbar punctures. It provides a very big benefit to many SMA patients. However, there's a few caveats that I've detailed a bit more, I think, in the next slide. So after Spinraza was approved, this lumbar puncture issue, for instance, provides a significant logistical issue for clinicians and people that have to administer it. Spinraza is required to be administered to SMA patients repeatedly, particularly in the beginning. And so it meant that suddenly certain neuromuscular clinics were spending almost all of their time on administering this therapy, whereas they also had to do their other work, obviously. So that was an unexpected logistical challenge. The timing for Spinraza is very important. There's a limited therapeutic possibility where Spinraza has a maximum effect. Unfortunately, also, the pharmaceutical company that developed this drug charges a very, very large sum of money for it. And in fact, what happened here in the UK is that NICE in England decided, based on the cost versus benefit analysis that they did, to not recommend Spinraza for treatment on the NHS, just because it's too expensive. In contrast, in Scotland, the Scottish Medicine Consortium decided to approve Spinraza for very young patients only, and not for the other SMA patients. So that's really good news and really positive, but with considerable limitations at the same time. Fortunately, a number of other therapies are also in development, both therapies that target this SMN gene, but also therapies that are aimed to stimulate muscle function, and other various therapeutic targets that increase the overall health of motor neurons. And what I think is personally exciting is this prospect of combining different therapies for SMA. So both those therapies like Spinraza that target SMN, but also some of these other therapies that are currently in development. And an example from some of our colleagues in Germany shows that in SMA mouse models that were treated with a Spinraza-like molecule, there's a modest improvement in survival, or roughly doubling of survival. So this line depicts SMA mice that were untreated. The black line it depicts mice that were treated with a dose of Spinraza that's comparable to patients. But then when they added onto this black line, which is, again, this is the Spinraza-like treated mice, when they added another molecule on top of it, suddenly the survival was increased almost tenfold. So I think this is a very exciting prospect, and that we'll see more about in SMA research. So some lessons. I think I don't have much time left, but I think this is important to discuss, that we can take from this for drug development for other diseases. Spinraza is not a drug that can be repurposed for other disorders. It's very specifically targeting one genetic defect. However, what can it teach us is how to target genetic defects in general. And the technology that's used for Spinraza is very likely to be applicable to other specific genetic diseases in the future. It also tells us some of the limitations of those approaches. Getting those drugs into the central nervous system is not easy. It's very demanding for the patients and also logistically for the clinicians that need to administer it. And those are things to keep in mind when developing these kind of therapies. Is the effort worth the benefit for the patients? The design of clinical studies is something I haven't talked about much. The successful clinical trials that led to the approval of Spinraza were preceded by a number of studies that were unsuccessful. And so although there was much disappointment initially around those unsuccessful studies, they actually provided very valuable insights into how to design a successful clinical trial for a rare disease such as SMA. And they led to the conclusion in the case of SMA, for instance, that it was very important to be very selective in the patients that were included in those clinical studies. So where there can be a desire for as many patients as possible to be included in clinical studies, it might actually be beneficial to be very selective in selecting a very homogeneous group of patients initially to improve the chances for successful approval of a drug, and then go on to treating larger groups of patients. And unfortunately, it also illustrates how approval of a drug doesn't necessarily immediately lead to a wide availability of the drug to all patients worldwide. And I think that's a very striking example in this case, how unfortunately Spinraza is still not available to all patients in the UK. So I think I'll leave it at that because I'm running out of time. I have some slides about basic science that I'm happy to talk to you about later if you want to. Some molecules and various experiments that I did over the years. But I'll quickly show you this, if I'm still allowed to, as some take-home messages. I think SMA is a good example of a disease with a very specific, well-characterized genetic cause. So it might come across as a very simple disorder, but in fact, it can be very, very complex to investigate and complex to treat. As I mentioned, at the last point on my previous slide actually, but I think this is super important. And ideally, I think this is discussed and thought about before a therapy gets approved. How, if a therapy gets approved, do we make sure that it gets to all patients and that all patients have access to it? This is the part I had to skip because I want, as an academic, I talk too much about too many different things, so I tried to squeeze in too much into the presentation. Still, very many research questions remain, and I think that the next few years, with Spinraza being approved and SMA patients being treated with Spinraza, will be a very interesting time to follow the developments in the SMA field. I have a lot of colleagues that I work with that I all like to thank very much, and funders, of course. We can't do science without funders. I'm personally funded by the Wellcome Trust, so particularly like to mention those. And I think I would be very happy to take some questions if you have some.